Hi, my name is Madison Ross. The date is May 23rd, 2002, and today I will be doing my presentation on the determination of gender and if it's genes versus hormones. So my first slide here talks about the generic chromosomal differences between males and females. Within females and males, we have a 23rd pair of chromosomes that you can see down here in the bottom of these karyotypes. And it's either XX, as you see here in females, or it is going to be XY that you see over there in males. Now, there are a couple different variations that can form. You can see XXY, which will code as a male. That presence of the Y will always give you the determination of male. Or you can see things like XO, which is what we call Turner syndrome, and this will code as a female, but because it will not have that second X, they will not have normal female reproductive functions. Another interesting fact about the females, as you can see here, there are two X's, and what they will end up doing later on down the road is they will actually condense one of those X's into a clump of nuclear chromatin, and they will call it a Barr body. It becomes completely inactivated, and all of the genetic material actually will get pulled from that second X. Females will receive one X from each parent. You'll get an X from mom, and you'll get an X from dad. And very similarly, in males, they will get an X from mom, and they will get that Y from dad. They will not be able to take the X from him. And that Y chromosome is going to be what is essential for the development of our male reproductive organs. It carries a very specific gene that we'll talk about in a little bit that codes for being a male. Now, this second slide here, again, just talks a little bit about our chromosomal determinations. So right off the bat, you'll notice from the bottom that there's a 50-50 chance genetically of having either a male or a female. It doesn't always seem that way, but when you look at the genetics, it is. Just a fun thing to note is that reproductive structures in children actually do not start to grow inside of the baby when it's still in the mom's stomach until about 7 weeks of development. So until then, there's no distinctive morphology, and the morphology is defined as bipotential, because meaning two potentials, there are two possible ways it could still split. So once the chromosomes split, they randomly combine in order to create a zygote with the newly assigned gender. And again, that won't show until 7 weeks. But you can see here we have the parent's phenotype, or how they physically present, and we have our female and our male. And then after that, we have our XY, and we have our X, which are the parent's genotypes, or how they genetically are coded. They will split into the gametes, and you have, again, mom's two X's and dad's X and Y, and they randomly combine. You can see all the different possibilities of how we end up with our 50-50 chance of either having a female or a male. And again, genotypes, which are going to be these, will be how we genetically code, and the offspring's phenotype would be how they physically present. Now, our third slide is going to talk about the early stages of development, and specifically this slide is going to talk about males, and we will talk about females a little bit later on. There are two main pairs of accessory ducts for internal genitalia in said bipotential genitalia. Like we mentioned earlier, that bipotential means it's not sure yet, or it is not far enough along yet to present as either a male or a female. Now we have these Wolffian ducts, which will form out of the embryonic kidney and form the epididymis, the vas deferens, and the seminal vesicle in the presence of testosterone. And obviously that will form into what we would know as a male. And the Mullerian ducts would form what we would refer to as a female, as they become the fallopian tubes, the uterus, the cervix, and the upper half of the vagina in the absence of AMH. Now that AMH is anti-Mullerian hormone, which is what's produced when the Wolffian ducts take precedent, and it will actually cause the Müllerian ducts to degenerate, and they will not form into anything. And that is also what will happen if we have a female develop. The Müllerian ducts will develop into what you can see there, and the Wolffian ducts will degenerate as well. And that's just what it says here at the bottom. As one set develops, the other one degenerates based off of available hormones and hormones that are now being produced. Another thing about them is that our bipotential gonad has both an outer cortex and an inner medulla, and in females, our medulla will regress, and the cortex becomes what we know as our ovaries, which is the female gonad. And the cortex regresses in males, while the medulla becomes the testes, which is going to be the male gonad. Following that up, we have the SRY gene. Now that's that gene I mentioned at the very beginning that sits on our Y chromosome, and again, because it's called the sex-determining region of the Y chromosome. And this is what makes males males. Now, in the presence of this gene, the bipotential gonads, like I mentioned before, this is the hormone that will allow them to develop into testes. Without it, they develop into ovaries, and that's going to be the separate part of the gonad. Now, the SRY is going to produce the testes determining factor, now TDF, which will bind to the DNA and activate other additional genes to be produced for that male phenotype and that male genotype. Then, once they are finally differentiated again at that 7-week mark, the external and internal genitalia will begin to develop and secrete hormones, or in the case of the males, we have the androgens, which is the DHT, or our dihydrotestosterone, and testosterone. They're two separate things, and they are the two major steroid hormones in males. Now, the very interesting thing is that testosterone is not actually required for the testicular development. It will not be secreted until after the gonads actually differentiate into said testes, and it is recognizable as a male. Now, this picture here is what we're going to use to talk about our female embryonic development. When I mentioned before that you have your bipotential things, you can see up here that this would be what would be known as our bipotential gonads and things as such. You have your urethral folds, you have your labioscrotal swelling, which are going to be these purple ones out here, the genital tubercle, and our cloacal membrane. Now, as those different hormones start to be present, you will see the splitting where we'll either go one way or the other, and we will start on the male side since that was what we've already discussed. You can see the male developing the glands of the penis as they start to form out, and you can see as you go further down that this is what will develop, which is what we know as a stereotypical male. On the other side, though, you can see obviously we have what we would define as a stereotypical female, but they both start out in the same way. Again, here it's just important to note that the females will inactivate that X, that second X, and the either male, maternal or paternal, the other X will take over the dominance. There's no SRY gene that is going to be produced here, so the bipotential gonad forms ovarian tissues instead, which is what will start our female development. There's also no AMH, no anti-Müllerian hormone, so the Müllerian ducts will start to create those fallopian tubes, the uterus, and the upper half of the vagina, and the Wolffian ducts will degenerate without the testosterone and the DHT, leaving us with the presence of all of the female external and internal genitalia. These are my references. I used the textbook as it was a great resource for this project. That is all I have for you. I hope you feel like you learned something, and thank you very much.